Lice genes offer clues to ancient human history. The jumpy parasites have followed our ancestors around for at least 25 million years, adapting along with us through major upheavals. Along our evolutionary journey from monkey-like primates to bipedal apes to big-brained humans, we have had the company of an extraordinarily loyal companion, Pediculus humanus, otherwise known as the human louse, and all the while, the lice have recorded this journey in their genes. A new study, for example, found that some lice in the Americas are hybrids of those carried there by Native Americans and others ferried across the Atlantic by European colonists. "We humans do not live in a bubble," said Marina Eskuntz, an evolutionary geneticist at the U.S. Department of Agriculture and an author of the new study. Lice are part of our lives and our history. Lice commonly dwell on people's heads, clamping onto hair shafts, piercing scalps and drinking blood. Unable to survive away from human bodies, the parasites jump from one person to the next. If people are crammed together in unsanitary conditions, such as an overcrowded jail, lice can spread into clothes and feed on other body parts. Other mammals and birds have lice of their own. Each species of the parasite has exquisite adaptations to its particular host, be it a penguin or a bat. This intimate association is ancient. In Germany, paleontologists discovered a 44-million-year-old louse with bits of feathers preserved in its gut. Lice fossils are too rare to reveal very much about their history, but their DNA contains many additional clues. By analyzing genetic material from lice, entomologists can build their family trees, revealing which species are most closely related. Often, the closest relative of a louse species lives on the closest relative of its host. For example, in the early 2000s, David Reed of the University of Florida and his colleagues found that human lice are most closely related to lice that live on chimpanzees and more distantly related to lice that live on monkeys. For some 25 million years, in other words, our lice have been following us down our evolutionary path. That's not to say that lice are completely loyal. Another species, Thrus pubis, better known as crabs, dwells only in human pubic hair. Crabs are not closely related to head lice. Instead, Dr. Reed and his colleagues have found their closest cousins are lice that live on gorillas. It's possible that early human ancestors picked crabs up while sleeping in an old gorilla nest or feeding on gorilla carcasses. In another provocative study, Dr. Reed and his colleagues compared human lice from different parts of the world. They looked at genetic material known as mitochondrial DNA, which is passed down only from females to their offspring. The researchers found that many lice belonged to one of two lineages. Remarkably, those lineages split from a female louse that lived perhaps 1 million years ago. Dr. Reed and his colleagues speculated that this deep split came about when humans expanded out of Africa. Along with their own lice, they picked up lice from Neanderthals or some other extinct group of humans. More recently, lice researchers have turned their attention to the chromosomal DNA that lice inherit from both their mothers and fathers. In 2010, Dr. Eskuntz joined Dr. Reed's team and led an effort to collect such DNA from a broader sweep of the world. In the new study, published on Wednesday in the journal PLOS One, Dr. Eskuntz and her colleagues analyzed DNA from 274 lice collected from people in 25 places across the world, including Honduras, France, Rwanda and Mongolia. The DNA revealed 2 geographic clusters of lice. One was present in Africa, Asia and the Americas. Among these lice, the researchers found a close genetic link between Honduras and Mongolia. They suspect this kinship is a sign that the Asian people who first spread into the Americas about 23,000 years ago brought lice with them. The remaining lice formed a second cluster, which the researchers found in Europe, as well as in the United States, Mexico and Argentina. The researchers also found 33 hybrids of the 2 clusters, 25 of which lived in the Americas. Dr. Eskuntz and her colleagues see in these results a chronicle of modern history. European colonists sailed to the New World, bringing their lice with them. In the Americas, the second cluster spread and sometimes ended up on the heads of people already infected with lice from the first cluster. But if these lice are indeed colonial hybrids, Dr. Askuntz and her colleagues are puzzled that they haven't found more. The rareness of the hybrids might be the result of some kind of barrier to interbreeding. It's possible that the two clusters of lice were isolated from each other for so long that they gained mutations that didn't work well when they were mixed back together. Dr. Askuntz said that lice researchers are only at the beginning of their work. In the new study, she and her colleagues looked at just 16 small regions of louse DNA. The next wave of research will examine the entire louse genome, and she expects this new data will yield more insights. It may be possible, for example, to understand how human lice evolved the ability to move from the head to the body, and why only body lice carry microbes that can cause diseases such as typhus. And researchers may be able to pin down exactly how our ancestors picked up the lice that still bedevil us today. The genetic information we are seeing in current human lice can still tell us things about our human past, said Dr. Askuntz. The human louse, Pediculus humanus, is an obligate blood-sucking ectoparasite that has co-evolved with humans for millennia. Given the intimate relationship between this parasite and the human host, the study of human lice has the potential to shed light on aspects of human evolution that are difficult to interpret using other biological evidence. In this study, we analyzed the genetic variation in 274 human lice from 25 geographic sites around the world by using nuclear microsatellite loci and female inherited mitochondrial DNA sequences. Nuclear genetic diversity analysis revealed the presence of two distinct genetic clusters I and E, which are subdivided into subclusters, EIB and IEIB, respectively. Among these samples, we observed the presence of the two most common louse mitochondrial haplogroups, A and B, that were found in both nuclear clusters I and E. Evidence of nuclear admixture was uncommon, 12%, and was predominate in the New World, potentially mirroring the history of colonization in the Americas. These findings were supported by novel DIB simulations that were built using both host and parasite data to define parameters and models, suggesting that admixture between CI and CAE was very recent. This pattern could also be the result of a reproductive barrier between these two nuclear genetic clusters. In addition to providing new evolutionary knowledge about this human parasite, our study could guide the development of new analyses in other host parasite systems. The human louse, Pediculus humanus, Thyroptera Anoplura, is a blood-sucking, wingless, human-specific ectoparasite that is obligate, cannot live without the host, and permanent, complete their life cycle on a host. This relationship has made humans and lice ideal for coevolutionary studies. In the past, studies have shown that primate lice co-speciated with their hosts over the last 25 million years. Furthermore, phylogenetic analyses revealed that human and chimpanzee lice in the genus Pediculus diverged ca. 5.6 mi ago, which is contemporaneous with the known divergence of chimps and humans. Human head lice have been found in archaeological sites, including a head louse knit obtained from human remains in northeastern Brazil dated 10,000 years old, and a knit from Israel dated 9,000 years old, among others. Multiple archaeological findings of human lice around the world are shown in supporting the idea that lice are one of the oldest known human ectoparasites. In host parasite systems, these long-term associations could lead to reciprocal adaptations in the hosts and parasites, i.e., coevolution, which allows one to build phylogenies for both the parasite and host and to obtain time estimations of events that co-occurred in both the host and parasite. In cases where the parasite and host have an inextricable relationship, the parasite phylogeny could reflect host evolutionary history. In some cases, there are also parasite-specific life history traits such as relatively faster evolutionary rates and larger population sizes that allow parasites to maintain greater levels of genetic diversity over time than their hosts. This allows researchers to harness the parasite's genetic diversity in order to explore unclear aspects of host evolution based on direct host evidence, such as archaeological remains or host molecular data. Evolutionary studies of human lice have already proven themselves as a useful tool in human evolutionary studies. They have provided insights on the ecology of early humans, such as when humans started using clothes and how humans traveled globally. Recently, human DNA was extracted from nits found on hairs from ancient mummies in South America, revealing ancestral human migration routes. Moreover, almost 20 years ago, Reed et al. Described highly divergent human louse mitochondrial clades, hypothesizing that each louse mitochondrial lineage evolved on different hominin hosts. The authors suggested that as anatomically modern humans migrated out of Africa and interacted with other hominins, direct physical contact among hominins allowed different lineages of lice to be transferred from host to host. The hypothesis of direct contact between modern and archaic hominins predicted using human louse data was later confirmed through host general